Hi everyone, I'm Rachel. I'm a second year PhD from Dartmouth. I'm really excited to be here, and I want to say thank you for organizing this great event as well as giving me the opportunity to talk today. So my presentation today is on systematic benchmarking of imaging spatial transcriptomics products in FFPE tissues. So spatial transcriptomics, as you guys probably all know, rapidly evolved in the past few years. It's a technology that essentially measures the gene expression profiles of tissue or cells in cyto. So these approaches have overcome the limitations of single-cell RNA sequencing by negating the need for cellularization and maintaining both local and global spatial relationships. Spatial transcriptomics addresses another limitation for single-cell RNA sequencing, which is to work with formalin-fixed, paraffin-embedded, which is known as FFPE tissues. FFP is the standard format for many clinical sample preservation, as well as for pathology, due to its ability to maintain tissue morphology and sample stability for decades. And the ability to process these kind of samples would enable the use of archival tissue banks for studies and obviate the need for specialized sample harvesting workflows. So to summarize, spatial transcriptomics have really provided researchers with the opportunity to kind of advance therapeutic approaches to investigate these complex biological processes and essentially develop novel diagnostic methods and many, many more. So, three companies recently released FFPE compatible commercial platforms. They are 10x, Visgen, and NanoString. So, spatial transcriptomics methods mostly use variations of fluorescence in situ hybridization, known as the FISH method, where the mRNA molecules are tagged with hybridization probes, which are detected in a combinatorial manner over multiple rounds of staining with fluorescence reporters, and then imaging, and then washing off and restaining. So the main differences basically lies in the transcript amplification. So Xenium from 10x uses a way which is called the rolling circle amplification. Cosmics from NanoString, on the other hand, uses a low number of probes amplified within like kind of the branch chain hybridization. And lastly, Merscope from Visgen uses direct probe hybridization. And then computational reconstruction yields the map of transcript identity with the single molecule resolution. So now the question kind of rises, how does the performance of each platform compare? Okay, well, you see it now. And three companies provided in total of 6 panels for customers. 10x Xenium provides 3, human breast, human lung, pan-human. Visgen Merscope has 2, human breast and human lung, and the NanoString Cosmics only has 1, which is a 1,000 geneplex pan-human panel. So in this study, we essentially prepared 2 tissue microarrays, one with 7 different tumor types, another one with 16 different normal tissue types. And with this head-to-head performance comparisons, we hope to understand the key differences across platforms, which will allow us to make better informed decisions regarding panel design, method choice, sample selection, and many more. And now I will go through the results in 3 comparison levels, which are transcript, tissue, and cell. So for transcript level, essentially the question we're trying to answer is, who detects the most transcript with accuracy? So here at the left, you see a heat map of total transcript per gene across tissue types. Xenium generally detects the most number of transcripts for all tissues and cancer types, and tissues such as spleen, lymph node, thyroid have an unsatisfying results across all panels. And on the right, we have dot plot that compare the number of transcripts for the overlapping genes across all platforms. Xenium breast has 15-fold more counts than the Cosmics multi-tissue data, and the Xenium multi-tissue panel has a 12.3-fold higher expression, Lastly, having a 14-fold difference with the long panel. Merscope also shows slightly higher expression level than Cosmics for the breast panel, with a fold change of 5.4. And finally, Axenium outperforms the long panel when compared to Merscope and has a comparable expression level for the breast panel. So take-home message, Axenium detects the most number of transcripts. But that's not the only case we want to consider. Next, we want to assess the specificity of each platform. So Xenium and Cosmics, they include both negative probes, which are essentially real probes targeting nucleic acids that are not present in human tissues, and negative barcodes. There are barcodes that are not associated with any probes in the experimental panel, whereas Merscope include only negative barcodes. So that will actually allow us to calculate the fraction of negative barcodes and probes relative to the number of transcripts for each tissue type. So what we observe is that Xenium and Merscope have high on-target calls, while the performance from Cosmics was not as satisfying. Then by looking at the false discovery rate, we have found that actually Cosmics suffers from really high FDR. And lastly, by evaluating the number of genes reliably detected by calculating the percent gene above noise, we can conclude that Cosmics has low number of genes above noise, high FDR, in consequence, low specificity, despite its panel having 1,000 genes, which is a much higher number than the other platforms. And to conclude the transcript level, Xenium typically shows highest transcript counts without really sacrificing any specificity. Then moving on to tissue level. Since it's difficult to evaluate whether a high number of expressed genes is actually representative of increased sensitivity to real biology or increased false positive rates. So we evaluated the correlation of our data set to reference RNA-seq database. What we did was we aggregated pseudo bulk tissue TME results from all panels and compared them to TCGA and GTEx, which are two really well-established databases out there. So Xenium overall has the most consistent performance across all TMAs and panels, yielding correlation around 65 to 80%. And then looking at Cosmics, the upswing character that Cosmics shows at the lower gene expression and is consistent with actually the low number of genes above noise and the high FDR rate that we just mentioned. And then last but not least, Merfish has really unstable performance, showing comparable correlation, but only with certain tissues. We also noticed that the unsatisfying Merscope long panel performance, while with extremely low number of transcripts detected, that actually might be due to the experimental workflow. Since 5 nanometer imaging was used, we are reperforming experiments with 10 nanometer. But again, Merfish still suffer from a low correlation with many tissue types. And next, to further analyze whether the results match previous biological knowledge, we determined the expression of tissue-specific transcript markers and curated tissue markers from GTEx and TCGA databases. So here are heat maps from the 3 panels with all the normal tissues as rows and tissue markers as columns. So we are expecting a ladder-like expression pattern here. As you all can see, Merscope show expression in some markers, but not all of them. And then although Cosmics show satisfying expression patterns for some tissue markers, for example, those, but we observe that many canonical markers are not enriched in expected tissues, which may be again caused by the high FDR and low sensitivity. And in conclusion, actually, Xenium has a really distinct expression patterns, whereas Cosmics and Merscope show a less distinct pattern for tissue-specific expression. And then moving on to cell level, we want to ask who performs the best in downstream analyses. So we chose a permissive threshold for removing cells with fewer than 10 transcript counts for Xenium and Merscope and 20 transcript counts for CosMx. The tumor TMA constantly had a greater fraction of cells passing through the filtration for around 90%. And the normal tissue TMA had overall lower cell retention performance. But the relative performance of the platforms based on the fraction of cells basically remained the same. So CosMx and Xenium basically retained in total around 77 to 80% of the cells, and Merscope data performing less than that, but still have a reasonable amount of cells retained for the tumor microtissue, tissue microRNA. And then we want to perform clustering, which is a commonly used downstream analysis in many sequencing data sets. So here, Xenium in the first row resulted in total of 9 cell types detected for the breast cancer tissues that we selected. On the other hand, CosMx resulted in 8 tissue types on the second row. And resulted in 6 cell types on the third row. The cell type annotation of Xenium and CosMx is pretty comparable in terms of both the transcriptomic profile and the subtype depth. CosMx only unable to annotate 2 immune cell subtypes, which are B cells and T cells. And gene expression of the same cell type from both platforms correlate well, as we can see here from the heat map on the right. The cell type annotation of CosMx, however, we want to mention that it was especially difficult compared to Xenium and Merscope because of its atypical gene markers shown for each cluster in the heat map and low expression of transcripts from some canonical markers that were missing that we showed previously. And lastly, Merscope identified most but not all cell types recognized as Xenium and CosMx. Merscope and Xenium, however, showed a really strong high correlation for almost all matching clusters. And we can see the heat map on the right corner shows a clear one-to-one mapping between the Merscope and Xenium clusters. So to summarize, there are a lot more that was not mentioned, including the cost, experimental workflow differences, process of data acquisition, cell sublimation pipeline, many, many more. And in short, to conclude, we think that the amplification of RNA signal is essentially important for recovery of transcript counts by spatial transcriptomics technologies, especially in some low-quality samples where RNA might be highly degraded and fewer landing sites are available for probes, which is the case now because platforms such as Xenium rely on small numbers of landing sites and may subsequently heavily amplified, so is robust to this RNA degradation, so is more broadly compatible with a broad range of samples. So here in this presentation, you might get the sense that Xenium outperformed the other platforms in some cases. On the other hand, when sample quality is high, the gap between amplified and unamplified platforms is much smaller. And we think that most platforms can yield useful data for subsequent downstream spatial analyses. So one sentence. So choose what's best fit expectations and answers to questions. Every platform has its up and downs, and that's the best summary that we can get. Some next steps. So we're looking for more experiments, especially for validation experiments, and reperform some Morphish runs for 10 nanometer imaging step. And we're looking into also more data analysis and downstream analyses. I only have 15 minutes, but this paper is now on bioRxiv. So if you want to look into it and essentially dig out the parts that I did not have time to mention in this presentation, please feel free to do so. And last but not least, I want to thank my lab members and a lot of my collaborators from Broad, Harvard, and also the 3 platforms, as well as the funding sources. Thank you so much.
time for one question, and then we'll have a panel session for other question. Okay, one question. Okay.
So throughout the talk, I got the impression that Xenium came up as as the best among the three. And in the end, your conclusion was choose what fits. It was a non-committal conclusion. Is there any particular evaluation where Cosmics was clearly the best?
Cosmics was clearly the best. I mean, yes, it has 1,000 genes. Cosmics, right now, Xenium can only has up to 300-ish genes in its probe. But I want to also let everyone know that 10x claims that they're having this 1,000 gene panel coming out in half a year, probably end of 2024, start of 2025. So we'll see if that's the case. But right now, Cosmics has up to 1,000 panel gene, which is valuable. So, yeah, the short answer is yes, of course.